Hey everyone, welcome to the Being Patient podcast. I'm Deborah Kan, founder of Being Patient. When my mom was diagnosed with Alzheimer's disease, I decided to use my skills as a journalist in a different way. Frustrated by the lack of information on science and the inability to get different expert opinions, I decided to quit my job at the Wall Street Journal to create a better platform for people impacted by dementia. We are a community where news and information is created by our team of journalists. We ask tough questions and we simplify the science so that anyone can understand. We don't only cover disease, but delve into the latest research on what it takes to keep our brains healthy. We invite the experts and ask your questions. Here's today's podcast. I hope you enjoy it. Okay, hello everyone. Welcome to Being Patient Brain Talks. I'm Deborah Kan, founder of Being Patient. Today we're going to talk about gene therapy as it applies to Alzheimer's disease. I know a lot of people in our community have said they're getting genetic tests for Alzheimer's disease. Well, genetics is actually a target for figuring out the mystery behind Alzheimer's. Joining me now is Dr. Ron Crystal. He's of Cornell University's Weill Medical College and joins us now from New York to discuss this topic. Thanks very much for joining us, Dr. Crystal, and welcome.
Thank you.
So let's just start at the beginning, because I know you were one of the first scientists to really delve into the role that genetics can play in terms of preventing Alzheimer's disease. Tell us how this all started from your personal experience.
Well, gene therapy is essentially a drug delivery system. It's just what we're doing is we're delivering DNA, genes, and we're all made up of about 25,000 genes, and we're using those genes essentially as drugs. So gene therapy is a strategy to try to prevent or treat disease by using genes as the drug.
You were not, were you in the field of genetics at this time? I think you told me you were researching another disease. So what led to kind of a focus on Alzheimer's?
Well, some colleagues of mine had gotten the idea related to what we are actually working on. And the concept relates to the genetics of Alzheimer's. So we know from epidemiologic studies that the apolipoprotein E gene is the important genetic determinant in terms of risk for Alzheimer's. And there are three forms of the apolipoprotein E gene referred to as ApoE: ApoE3, ApoE2, and ApoE4. Most of us are ApoE3. We inherit it from both parents, the ApoE3 gene, but some of us have the ApoE4 gene. And if we inherit that from both parents, we have about a 15, maybe a little more risk for the development of Alzheimer's. And when it does develop, it's more aggressive and it occurs earlier. In contrast, if you're lucky and you inherit from your parents the ApoE2 gene from both parents, the ApoE2 gene is protective. What got us interested was the individuals that are inherit from one parent, ApoE4, and one parent, ApoE2. So they're what we call a heterozygote, ApoE2, ApoE4. And in those individuals, the ApoE2 gene takes away most of the risk of the ApoE4 gene. So we got the idea, why not modify the brain genetically of ApoE4 individuals or ApoE4 with the ApoE2 gene so they would have lesser risk for the development of Alzheimer's.
So, I mean, the concept of really altering your genes to make them behave differently, what does that exactly mean? Like, how do we change our genetic makeup in order to make a risk gene behave in a less risky fashion?
Well, the target of the brain cells, and so we're trying to get the proper gene into the brain cells. Now, we're not really altering the genes. What we're doing is putting in a new gene. We're augmenting the genes. And so what we're doing is taking the ApoE2 gene and delivering that to the brain. Now, one of the obvious questions is how do you deliver a gene? You can't just eat a gene and expect that it goes to the brain. And the way we do that is with viruses. There's a group of viruses. The viruses we use are called adeno-associated virus, or AAV. And these are been widely used in terms of brain gene therapy because they're very avid for brain cells. And the viruses are modified so they don't cause harm. We're essentially using the viruses like a Trojan horse to carry the normal gene, the ApoE2 gene, into the cells of the brain. The cells then will make ApoE2 and bathe the brain with the ApoE2 and hopefully convert the brain of the ApoE4 homozygote to that of an ApoE2-A4 heterozygote.
How is it, I mean, if the virus plays the host to be a delivery mechanism, how can it be lasting? I mean, is it, viruses come into our bodies and then we recover? Like, why would we think that would have a lasting impact?
That's a good question. So first, what we do is we engineer the virus so it can't cause harm. That's important, that we take out the parts of the virus that cause harm. The second thing is the way this virus works is it enters the cells, in this case in the brain, goes to the nucleus, that's where the genes are, and it just sits there. And it hijacks the nuclear machinery to make the new protein, in this case ApoE2. It just sits there. As long as the cell is quiescent, and our cells in our brain are quiescent, they're not turning over all the time. So as long as the cells are not turning over, it just sits there and functions forever. And at least in experimental animals, we know that we can genetically modify the brain for the life of the animal.
So at what stage are we in determining whether or not gene therapy will really work for diseases, neurodegenerative diseases like Alzheimer's?
Well, that's going to take a long time. Studies have just started in terms of the first thing to do is show that it's safe. The second thing to do is to show that you can actually get ApoE2 into the brain and get it to increase levels. And we started this at Weill Cornell in our academic program, but we can't do these big studies. We need a company for that. And so I was lucky enough to start a company called Lexeo Therapeutics, and they're carrying out the studies now in multiple sites around the country. So the important thing is first showing that it's safe. Secondly, show that you can actually get ApoE2 into the brain and that it will persist. And then the hard part is proving that it really works, which takes large numbers of patients, and that'll take several years.
Yeah, and I should mention that we're doing this talk in partnership with Lexeo to bring attention to gene therapy and explain to our audience really what is gene therapy when it comes to Alzheimer's disease. So I guess I have a question about the fact that, there are people out there who have Alzheimer's disease and they don't have the genetic link. So would this type of treatment really only be for people who have that genetic predisposition?
Well, there are really two groups of individuals related to ApoE4. There are those that inherit it from both parents, what we call the ApoE4 homozygotes. And there's about 1 million people in the United States that are ApoE4 homozygotes. So they're at the highest risk. But also at risk are individuals that are what we call heterozygote. They inherit one ApoE4 gene together with another gene like ApoE2, ApoE3, and can we help them as well? So we're starting with ApoE4 homozygotes, but if this works, then it could be expanded.
So does that mean what does that mean for, the majority of the E3 population? If we know we can make E4 act more like E2, which is protective, what does that mean for most people who are homozygous E3?
That's a good question. We don't know the answer to that, but it's possible that the ApoE2 could actually treat those patients as well.
So I have a question about ApoE2. I've met quite a few, and we have quite a few in our community of people who disclose to us that they have 1 or 2 copies of E4. And now in my years of focusing on Alzheimer's disease, I have never met a person with E2. I don't know if it's just because if it's not a problem, people don't talk about it. They're not, talking about Alzheimer's if they feel like they have extra protection. Do we know the prominence of the E2 population, and how much do we know about E2 in terms of the protectiveness factor?
The population of E2s are about 10 to 15%. So,
Versus
so
E4, which is E4 is what?
E4 is probably at 5 to 10%, something in that, and the E3s are the rest. So I've sequenced all my genes, and I know I'm an ApoE3. So as an example, so I have an average risk for that. In terms of how ApoE2 functions, ApoE has many different functions in the brain, and as you know, there in Alzheimer's, there is the amyloid accumulation and the tau, tau tangle accumulation. This doesn't pay attention to whether it's amyloid or tau because it's working at a more proximal level. So it's working at a level above that in terms of mechanisms of disease. So actually ApoE has many different functions in the brain, all of which relates in terms of improving amyloid accumulation, as well as tau and tau tangles, and a whole bunch of other mechanisms as well.
So it's also been described to me as kind of like the cholesterol transport gene. What is the interaction between that gene and the formation of plaques and tangles that puts people on what goes wrong and what makes you more susceptible if you have E4?
Well, that's a good and very complex question. So ApoE is made in the brain in glial cells, astrocytes and glial cells, and it forms the particles that carry cholesterol, which are picked up by the neurons within the brain, which have the receptors for ApoE. A lot of it has to do with the binding of ApoE to what is called heparin proteoglycans, part of our extracellular matrix. And we know that ApoE4 binds very tightly and forms a nidus for the development of amyloid and tau, whereas ApoE2 binds very little. And so that's part of the mechanism. But there's several other mechanisms as well, and that's still being investigated. There's papers coming out all the time in terms of why ApoE2 is better than ApoE4.
I want to talk a little bit about the actual research and, what Lexeo in particular is doing. So you said we have a lot more that we need to discover. I mean, presumably you're looking in this particular trial, you're looking for ApoE4 because you need 4 to do the gene therapy. But what is it that you, as a scientist, when you said you use a virus as a host, is that an injection? Is it a pill? Like how is this being tested on people who have a genetic predisposition?
So the question is how do we get the gene into the brain? And there are various ways to do it, but the most effective way is to go directly into the cerebral spinal fluid, that is the fluid bathing the brain. And so we do that with the what's called the interventional radiologists who take with local anesthesia in a CAT scanner. So the needle goes into right in the back of the neck, right where the cerebral spinal fluid is. Takes about 30 minutes to place the needle and a local anesthesia, and about 10 minutes to administer. It's an outpatient procedure.
So is it just a one-time procedure? It's not like, as other drugs where you have to go back every month?
If it works, one time.
So I'm assuming if that is the case, then why wouldn't it be that you could have the answers quicker? Because you have to wait and see if people actually come down with Alzheimer's. Is that right?
Well, the most important thing is say for any new drug, and this is a new drug, the most important thing, of course, first is safety. And so the initial studies are really safety studies, but also using biochemical markers to determine whether it works. So we sample the cerebral spinal fluid, the fluid bathing the brain. We sample that before and after, and we can measure ApoE4 levels, ApoE2 levels, what the ratio is, as well as some other markers as well. And so that tells us, in fact, are we making the drug, and does it persist, and are we making enough?
So are the targets people who have ApoE4 and plaque in their brains as kind of a marker as to these people could possibly be on the road to Alzheimer's? Is that
primarily individuals who have early cognitive decline, who are ApoE4, and also have risk factors for Alzheimer's, including amyloid plaque in the brain is a good example.
So can people with ApoE2 get plaque in the brain?
They can, but less commonly, and it's an older population and usually less severe.
So to monitor whether or not a drug like this will be effective, are we looking at the biomarker of plaque to determine that and understand if the person, I mean, there's people with plaque who don't go into aren't diagnosed with Alzheimer's disease in their lifetime. So
it's now possible to do radioactive scans to quantify the amount of amyloid within the brain, also the amount of tau within the brain. Both are being done in this trial.
Is the idea, though, to look for a reduction? Is it like some of the monoclonal antibodies that's trying to reduce the biomarkers like plaque, or is it just to monitor whether or not the brain of these people goes into kind of a disease progression?
At minimum, we'd like to make it stable and at best reduce it.
So I want to delve a little bit more into detail about what this means for the non-genetically predisposed population. Let's say that, I mean, there's other companies aside from Lexeo who are trying to use gene therapy for Alzheimer's disease as a target area of research. Let's say we do discover that gene therapy does indeed work on risky populations. How would that translate into perhaps a bigger solution for people who don't have a genetic predisposition?
Well, as I mentioned before, it is possible that ApoE2 could help that population as well. We just don't know. But other groups and other companies are working on multiple different ways. For example, growth factors that will stimulate neurons, brain cells to repopulate, remake more. So there's a whole variety of strategies that are being used. There are some cholesterol strategies that some investigators are looking at from gene therapy. So there's a number of different approaches for the population that are not ApoE4.
So Dr. Crystal, is this being used successfully in any other diseases aside from Alzheimer's? Is there something to compare this technique for Alzheimer's with another disease state?
Well, gene therapy started back in the late 1980s or so when we and others, many others, started the field. There are now some approvals for gene therapies. For example, spinal muscular atrophy, the SMA children, the floppy babies who now can sit up, and that's an approved therapy, for example. There is an eye disease that has been cured by gene therapy, a hereditary eye disease. And I think you're going to see over this year and over the next several years, several approvals of other gene therapies for other forms of disease. So we know that the technology works. The question is getting it right for the problem like Alzheimer's.
Okay, and I'm going to probably annoy you by asking this question because you said it, we're kind of a long way away, but I know people from our community will want to know, well, how long? Like, how long is it? Are we decades away? Or are we a few years away? Like, when do we hope to understand more? And I know a lot has to do with recruitment of trials, but, assuming that Lexeo or other companies out there do have enough participants, like how long do you think it will take to understand whether or not this is a successful application for Alzheimer's disease?
Probably these trials, the large phase 3 trials take several years, so probably 5-6 years before we know.
And if that is the case, the therapeutic, it's just a one-time, as you said, it's kind of, it has to go into the cerebral spinal fluid. So I'm assuming it's a shot or something in your spine. Is that?
No, it's a very simple procedure. It's done under imaging, so the needle goes in the right place. Local anesthesia to the back of the neck, and just a small needle that goes directly into the cerebral spinal fluid in the back of the neck. It's an outpatient procedure. It doesn't hurt. It takes about an hour, the whole procedure.
What is the risk to this exploratory work with genetics to the patient population?
Well, we carry out a significant amount of safety studies in experimental animals before we go to humans. So the regulatory bodies, the FDA, of course, require that appropriately. But you still have to know in humans whether it's safe or not. The viruses that we use we know are safe in experimental animals, but on the other hand, the immune system might be able to see the virus and create a defense against it. And so there's a whole variety of possibilities in terms of safety. So far, everything looks safe, but you have to do the studies to prove that.
Yeah, absolutely. Well, it's a really fascinating area. Thank you for your research. We look forward to understanding more about gene therapy. And it also really highlights how people who hold the Alzheimer's gene really do have a big role to play in determining how much more we understand Alzheimer's disease.
Well, absolutely, and it's really the individuals who participate in the trials who are the real heroes, because without them, we could not move along and develop drugs like this. And so I would encourage people to think about participating in trials. These go on all over the country, and it's really the participation of people with Alzheimer's that make this possible to be able to develop a cure.
Do you know any other ways of determining, I know this particular with Lexeo that you've been involved with is taking E4 and trying to make it behave more like E2, but are there other methods of gene therapy out there that you're aware of in order to, as cure, prevent Alzheimer's?
Well, there are certainly strategies. Another strategy, for example, is try to reduce the ApoE4 in the brain. That's another strategy. Then there are strategies in terms of many pharmaceutical companies are working on in terms of monoclonal antibodies directed against amyloid, against tau. These also could be delivered by gene therapy. There's work going on to see whether or not, instead of direct administration to the cerebral spinal fluid, can you give it intravenously? And we'll go into the brain. So there's a lot of work that's going on throughout the world in terms of this, with a lot of attention to gene therapy. As I mentioned, gene therapy is essentially a drug delivery system. What we're delivering is a gene. And the technology has been developed over the last 30 years and is now coming to fruition that really is beginning to work in many diseases.
Yeah, absolutely. Dr. Ron Crystal, thank you so much for your time and your knowledge. We definitely want to stay abreast of the latest information on genetics. We know, as I mentioned, a lot of people in our community have found out their genetic status. So this is a really interesting topic to us. Thanks very much for sharing all of your knowledge with us.
Thank you.
So for more, don't forget to sign up for our newsletter on beingpatient.com, where we tell you about talks like these. And thanks again for our partnership with Lexeo Therapeutics for this discussion. Thanks a lot, Dr. Crystal, and we will talk to you next time.
Thank you.
Well, I hope you enjoyed today's podcast. For more information on upcoming interviews, don't forget to sign up for our newsletter at beingpatient.com. That's beingpatient.com. And send us any feedback you may have, whether it's someone you want us to interview or any comment about our podcast series. You can do so by emailing info@beingpatient.com. Thanks for listening, everyone. I'm Deborah Kan.